Everybody more or less settled? Okay, so of these three, each table, so each of the three tables has 8 tubes already labeled and sitting in some type of holder, okay? Each of these 8 things corresponds to the 8 things you see on the board. Because they're very small, I had to abbreviate some of them, and so the green on the right side is what is actually on your tube, and the black is the full name, so you can follow on your procedure as well, okay? The calculations in number 2, the correct answers are on the board. I think previously there was like, we forgot the puck vector component, but please make sure those that I usually also calculated. Just so you all have an idea of how it's going to run. We're going to go through the PCR protocol. After you all have completed that, your group has completed that, you will bring it to me. I'll put it in a thermocycler. We're going to wait until all 11 groups are done, or 12 groups maybe, are done with the thermocycler, or done with their tube to start the process. Once we've completed that, we will go on to the restriction digest protocol. That should take maybe about 20 to 30 minutes. And then there's like some incubation. So the PCR will take about an hour and a half. Restriction digest takes at least an hour. During the restriction digest downtime, after we're done with that, we're gonna have a short lecture with Rachel. After that is completed, we will go through the final steps of actually running the gel. Okay? If you have any questions at all, please just find one of us and ask, all right?
I just want to review, so we did pipetting a couple of weeks ago. So how many stops are there on the pipette? 2. And where do you go when you're drawing up your liquid? What do? You go to the, what stop first? The first stop, right, then you put it in the liquid, you draw up slow, and then you push it all the way to the second when you dispense. Some of you saw this last time, but these are really, really small volumes we're gonna be pipetting. So, how do I know what range my pipette will work at? It's on there, right? So the lowest number, the reason why we had a multiplier on all these reactions, so you guys have, so everyone, like everything, the minimum you'll draw up is 2 microliters. So, but that's a tiny, tiny drop, if you haven't drawn up that small drop before. I have a question for Mike. Did you have the thing with the tubes down or no?
Yes, we
Yeah.
do. There's one next to the vortex, so yeah.
And then we'll spend these little tubes if we get them on the side.
Sure, I can engineer something.
Yeah, we'll see if we get some on the side.
Yeah, we'll find a way to,
You
if
won't be able
necessary.
to really see this, but I want to just give you guys a couple of little tips. So you definitely will want to keep your tube so you can see it when you're doing these small volumes. You don't want to have it in your holder where you can't see, because this is such a small drop, you won't know if you actually drew it up. I push to the first one, I go in, and you won't be able to see here, but it's such a tiny, tiny, tiny drop. And then a way I do this, you hold your little tube up, and then you want to get, kind of push off the tip onto the side, down low, and you can see your little drop come out. And you'll get used to seeing what like a 2 microliter drop looks like, or a 1 microliter drop. So we do have, because you guys will be sharing, so you'll want to take turns doing your actual reaction. But we have extra tubes on all the tables, so on downtime, you guys can practice doing those small volumes without getting a big bubble in it, and making sure you can see it. This is kind of a training for the eye. And then,
One other quick thing, please make sure you're following the order of the procedure, right? So there's a reason we add the water first, because the largest volume. It's much easier to pipette small volumes into bigger ones than vice versa.
yeah.
So please just make sure you add the water first and then subsequent steps after that.
And you don't want to repeat your tip, right? Because we're all using the same stock. So we don't want to contaminate our enzymes by accidentally mixing in like a forward and a reverse primer, or mixing in an enzyme into our plasmid. So if you think your tip has touched anything except for the inside of your clean tube or your tube where you're pulling out reagent, just dispose it and get a new tip, because tips are much cheaper than failed experiments. So.
Sure.
And I just saw yours says Rachel, so I'm just.
Oh, really? It was too small, so I did it one.
Okay. So are we gonna have them just start following their?
Yep. Yeah,
Sure.
that'll work. Just a reminder as well, the tubes, right? So there's 8 tubes at each bench, so make sure we're sharing. as well, the number is written on the cap and on the side of the tube. Please make sure you're putting the correct cap back on the tube when you're done.
All right. So yeah, so we'll have one of each of us at the bench. You have a protocol in front of you, and it's like a restaurant. How many of you cooked before? Yeah, most of you have cooked. Good. So doing lab experiments is a lot like cooking. You're just following the recipe step by step. So one of us will be at each bench to help you guys as you're going through. Get started. Let's do some science.
We have larges though.
The lab pen, right? So why don't we each bench like pick a letter that's going to represent your group. So what letter do you want? A. And why don't you just write it up here so we don't forget? And what letter do you want? Give me any letter. B. All right. D. D. Okay.
I'm waiting.
An X? Okay, perfect. So you want to label the top of your tube with your letter, and then let's also, whoever thinks they can write the smallest in your group, on the side of the tube write like up high PCR, because we're going to end up with 2 different tubes so we can keep track of it. Yeah, you can write PCR and your X or your letter as well. Yeah, just so we can identify whose tube is whose. Yeah, the other one's going to be during the restriction digest, so we can label that one later.
So if you have not done so already, please choose some either number or abbreviation so that we know which tube is your group's.
Really good.
You're each going to have 2, one for PCR and one for restriction digest. So whatever you choose, just make sure you put it on your protocol so we can see exactly. So you need to be able to distinguish PCR from restriction digest and your group.
So then the next time, hopefully I can run through it by myself without my friend. So they calculate the letter range and for a whole 100 microliters for each tube, satisfying the following conditions. So, do they need this?
Oh, you're not supposed to.
So the things that they've already done is calculations. And I just confirmed on the board. I thought previous.
Okay. Does this look familiar to you guys? Okay. So what's the total final? If final reaction's 100, how, like,
100
what's
microliters.
the...
And each amount is on the board A through G.
Okay.
So the first, yeah, so I recommend filling it out on the protocol as well, so it's easier to follow along.
This is one of your water. And how do you guys do this already? Okay, okay. And so this was that, like, CI. Okay, perfect. So 61, and then should be 10 for me. And then...
Has everyone written in the values on the board in the protocol?
Yeah. The bad news is we only have 2 small pipettes.
Oh, there
We should have 3 or 4 of the pipettes that we can water. But let's get started with the water. The water, there's one for each of you in these tubes. And this is special water. This is molecular grade water.
So the super tiny ones only work for the P2s. Most of them are going to use the bigger tips.
It's not just RNAs. And we use the term RNAs and DNAs. Does anyone have an idea what that is? A's? A's, yeah, A's is an enzyme you didn't need. The A's part indicates usually
Reach the top. It's not
that it's cutting up whatever you've done with it. So we don't want a lot
Yeah,
of
on most
foreign
pipettes, lines imply decimals.
floating around our system.
So
So there's all these RNAs and DNAs that get rid of that from our system.
And then it's 10.
So lots of these reactions will have
we end up having 12 groups, which I think should still be okay instead of 11, because like they're using each of the things.
oh yeah, oh
Like we
yeah.
didn't say to not use one of that,
Oh, yeah,
but
sure.
we still have enough reagents that we use.
Okay.
Where does the decimal start?
So, if the decimal starts, the black number is like that, and the
Red numbers and flags. I'm pretty sure I allocated enough volume, but if you get to one of the steps and one of the tubes not have enough of the reagent, just let me know and I'll get you more. You should have enough.
Deoxy mix.
There's a so there should be 2 sets.
Someone could have it. Where's the capital?
So why don't we do it this way? Who's done the B already? No one? You're on B. So when you're on B, then pass your hair and you can pass it.
So this is a P100. It doesn't go down to 2. Yeah, that's why it's starting to get stuck. This shouldn't go any lower than 10. Yes. Yeah, so you need either a P20 or a...
Yeah, so for small volumes, you want to put it into the liquid and put it down small times. Or actually physically touch it to get the volume.
Are we supposed to see it or
So you will, so how good your eyes? Have you done it yet?
Yeah.
Okay. Did you see it when it was here? No. Okay, so where's the water?
3? I'm looking. Do you guys have 3? Yeah. Yeah.
Or don't do it in there.
Sorry, where's the big thing of water?
Over here.
Here?
So you put it all the way into the water.
So this is more like you have to have really good eyes, but I know from experience this is going to go right to where the line is. And so, can you see through the tube right now? It's not rocket science. Do you see it? And now it's right at the line. And so when I'm doing this, I can actually see the line up and down. Right? So just be looking. So if you didn't see it, it's possible you didn't add it. So which reagent was it? Okay. So it's a smaller volume, so it's not working. So if you didn't see it, it's possible they're not there, so you can do that one again. It's not going to go wrong because you have too many potential ideas.
Wait, does that mean that
You're still missing it. So make sure you're all the way in the liquid. It's like go to the bottom of it and slowly drop. Okay?
So for groups of
Go
2,
to the very bottom of the tube and come up slowly. Like you should be able to see it right.
And then what?
I mean,
How are you?
And then when you're dispensing it, it's
Yeah?
there's a paper you see and then you do it.
That explains why you weren't seeing anything. It's alright, we're all good.
2 to 20, all right.
For some reason this one.
No, it's
We're here, yeah.
Rich is that 4 over there?
right here.
Can we get number 4? Okay.
Oh, are you guys ready?
Yeah, we're ready.
Number 4?
Yeah.
Oh, really? Okay.
Yeah.
Actually, I'm
We
gonna.
did A, B, C, and X.
Okay. Did you have one that says like PCR, one that says restriction digest?
Yeah.
Okay.
So 10
I think this is right.
at the bottom.
Yes, it'll be 1, 0.
2 microliters. Yeah, so you go to the first off. So you over here, the tension there, and then you press it down.
Here you go.
And then you
I meant to ask you too. So Aaron and I are meeting on Tuesday to hash out schedule, and I have a list of all the things I want to ask her about that, including when he would ideally like you to join us.
you'll feel it.
So how's the best way? I would say we could meet if you want. I could text you, email you, but basically I'm just gonna try to get some dates from him, and then I'm trying to do that.
Either will be fine. I think Tuesday is good. I could also come by and do that so this way you.
That's true, yeah. Maybe can I text you like Tuesday and see where you're at, and then we can figure it out. Will you be on campus?
You guys are also doing another nice thing as a group, and I always everything's lined up in order and you're putting it back in order.
Yeah, it has a location now.
Sometimes when you're working with you're like, where was I? So lots of times what I'll do is once I've done one, I'll put it down like one spot. So if I get mixed up, I know where I'm at.
Alright, so who lost the cat?
Sometimes I might be pipetting like
So, I'm sorry, unspecified. Puck 19 is the same as DNA template. If you see that in your protocol. So it's the same thing.
Yeah, but it goes.
Right, Puck 19 vector is DNA template.
Okay, open this one.
Okay, so 5 microliters.
So take it first off. Before you take it, first off. Then you put it. Now
What did you all label?
then you pull out.
YA?
There you go.
For both of them? So you should have two tubes, right? One for PCR, one for restriction digest.
Yeah.
So what's the name of your restriction digest? YA2. And PCR is YA1? AC? PCR? Okay.
Wait, like nothing's going in.
that's better.
Okay, wait, I'm pressing it now.
I think is everyone else done the template? Oh, okay.
Please don't. So don't do it so fast. Right. So once you get on one stop, go in, slowly come up. Very slowly.
Okay, wait, you try.
You're still not saying anything?
Yeah.
And there's liquid in the tube, right?
No.
Yeah, there is.
What is it? Is it set to? That's not 10. So the same way you're not saying anything. Everybody still good?
Yeah. Okay, YA1. Yeah.
Oh, they only have a bigger one. Let me see which one you guys are on. We're on. So you need 5 microliters. So what you can
That's
do
a
is-
P2.
Sorry?
They're-
So they don't have one that can do 5, so they could just do this 2, 2, and 1 out of it.
They don't have a P20 here? There's a P20 right here.
Oh, you guys have one? Oh, yeah.
Yeah, just you have to share your pipettes.
Yeah, I was saying
You guys go with my bigger sample.
What was your label?
So last time,
The letter B. Did you label your restriction digest one as well? Not yet. Okay. So, hang on. Let's wait. Because I don't know if you like 2 hours.
right size. For me, I always go in smaller.
You start that. Rachel's going to show you the vortex, and then come back to it.
Not good. Five is weird.
If it's 6, that's fine. Because you're gonna like potentially create bubbles if you try to put it back.
No, it's
did you guys all?
You're good. You're good. It's not gonna come out.
All right, so why don't we all?
It's gonna create bubbles.
Yeah, we have
So these are just really.
Cool. Make sure the cap is on tight and pick whatever costs after 6. I'll double check them all. And so
No, it doesn't
the
go.
polymerase. Those are 2 different things. Do you want polymerase? Do you want template? So that's Puck 19. So what's step 3? One. So template DNA was number 1. Puck 19. Did you already do that? So that was the very first one. Right. So. So yeah, so the order is not necessarily with that. That just tells you who you're choosing, right?
Okay.
So yeah, it should be Puck 19.
So we actually passed that.
So that was 2.
We need one. These
Yeah.
But you haven't added this yet, have you?
I mean, there's a chance we did it right, but
Because I definitely added it now for everybody. Okay. You're like do you have any remaining DNA polymerase, the red one?
And then, if you're working in a lab and
You're not dead, right?
working with scale, they'll have
The piece.
something
You added the red stuff? Oh! And so it goes at 94 for 1 minute, 45 for a minute and 30, and 72 for 3 minutes. And it repeats that 14 times. And that's how you program it, basically. This hasn't started yet. This is still room temperature. Once I close it and press start, it'll be running. No. So it's difficult. Keep in mind that the liquid is just a solvent, and so you're essentially putting a mass inside of it. So increasing the mass doesn't change the volume at all. If that makes any sense. But yeah, so it more like, if anything, it might evaporate a little bit if like the cap isn't on correctly because it's really like hot in there. But yeah, your volume will increase. Right now. What was your label again? I wrote it? Yeah, the Satan one, right? Yeah, anywhere you like. It's the red box. Because I know we should have enough of everything else, but that's the one thing I know. Everyone, please look up here for the next 30 seconds, please. So I'm going to start thermocycling on all 12 groups that have finished their first component. All right, just a reminder, the next step is restriction digest. You're going to be using number 1, 7, and 8. Right, so again, DNA template is the PUC19 vector. You're not going to be using the neb buffer, number 7. The restriction enzyme, the big one, number 8. All right. Just some general reminders. If for some reason you run out of a certain reagent, just let me know. I'll get you more. Please make sure whatever pipette you're using, you are not going outside the designated range. Okay. If you're using a P100 and it says 10 to 100, please do not try to pipette 2 microliters out of that. It's not precise enough. Okay. As always, if you have any questions, please ask one of the instructors in the room. Okay. Okay. Good to go.
Basically, anytime you want to
Yes. Thank you.
just cut out that small piece so that it'll really come off and then you want to amplify so you can do whatever stuff.
Also, for some reason, it says plasmid solution. That's the same thing as PUC19. I apologize for them being 5 different names throughout the protocol. But tray, please. Oh, wait. You have a question? PUC19. If you need more, I can get you more. But there was supposed to be enough, but.
Yeah. I'm going to have to know in all of my experiments about the temperature. Yeah, so actually you don't want to ever pipette with someone else holding it like that. It's hard. It's the same as trying to pour
Yes.
water with someone else.
Every table has 1 tube of each of those.
Yeah.
Wait, which one's the most one? Isn't
Oh,
the
yeah.
the same thing as
Yeah, so that just is a type, like a different fit on the tip. Yeah. So, and I mean, regarding, oh no, you're talking about sport. I think one is
what number is the buffer?
7. Right here. Right here.
I like the fit on those people.
The Gibco P2s don't eject properly, I think, with these tips. The blue ones,
Oh, okay. Alright.
Wait, I need
for some reason.
These ones?
No, my table has one.
Oh, okay,
Yeah.
okay. If, so,
The Gibson, sorry, not
so
Gibco.
if we get,
Do you guys
Okay, so please start over.
You guys have 8?
yeah, so you haven't done like 2 microliters and then some.
Somebody, it's still like,
So guys, when you're
I've
done
only
with your
ever
tubes,
ordered
put
from
them,
Sigma.
even if they're empty, put them on that
I
rack so
imagine
that
it
you
doesn't
keep track of them.
color-
This is a great way
Yeah.
to lose stuff and then to not know where it went.
Right,
So just make sure
exactly.
everything,
Yeah.
just put all of those, leave them on the rack with
Yeah.
the cap facing
Yeah,
up,
actually,
and double check that the cap.
I know for this one too, like, quick reminder everyone, your PCR restriction digest tubes cannot have the exact same letter or designation. So if you label the first one A, the second one cannot also be A. Make it AD or something. But PCR and restriction digest should have at least something different about them. Okay? RD, BD.
Still are working on the restriction digester. Everyone's done. Alright, so Mike's grabbing some water. So now we're gonna make our gels. And we don't have a whole bunch of gel boxes today, so we're gonna do the gels. We're gonna make 2 as a demo and show you how it's done. We're gonna need to make some buffer for it, so I'm gonna have you guys help me from last time figure out how I'm gonna make this buffer. So we're gonna make, this is TAE buffer. You use this when you run DNA gels. It's at a 10x concentration. And we're gonna need to fill our 2 gel boxes with it. So that's gonna take about 600 mils. And then we're gonna also need to make our 2 gels with this, which is gonna be like another 50 mils each. So we're gonna need a minimum of 700. So it'll be really easy to make just a mil of this, right? So I have 10x buffer, I want 1x buffer and water. How much of the TAE buffer do I need and how much of the water do I need? So you guys can jot down if you want to figure it out. So remember this is that CI, VI, CF, VF. So my final concentration is 1x and 1,000 mils or 1 liter. And my starting concentration is 10x, so I need to know how much of the TAE buffer and how much of the water.
So almost everything went in between 11 and 15.
All right, does everyone think they figured it out? So altogether, how much of the TAE buffer do I need? 100, right? Does everyone get that? And how much water am I gonna need? 900. Am I gonna use tap water? No. Especially because this reaction is gonna require polarity, right? So it's really important the concentration of ions in my water, positive and negative charges, and my pH. And that's really variable in tap water. So Mike is grabbing, or he has grabbed distilled water. And what am I gonna put in first? The water or the TAE buffer? Water, right? Because that's easier if I mess up to get rid of and get more. And what am I using to measure it? Right? Because that's my most accurate for large volumes. And what am I looking for when I pour this in? Besides not spilling it all over?
So I'll have you uncompile first, just make sure they understand what the purpose of this is, so you can actually see when you pull it out. Yeah. So it's like we're gonna put this in here, right? And so you actually have a colored background to see, so it's a contrast thing. But I'll let you put this together and make sure you use the 17 to the side. Yeah, so make sure the teeth are in. So it's not immediate, right? So you weigh this out. So it's 1 gram.
And I said to my group, but I got a really good question from my group about the restriction digest. So we all put it in at slightly different times. With restriction
Can you
enzymes,
reach the flask?
they have this, they can only cut in very specific places.
That's fine.
So once they've cut everything in your tube,
And so we'll start by adding
they're
50 mils of the
inactive. They won't
TE.
do anything else.
1x.
So with restriction enzymes, it's very important that you do the minimum time so that you can cut everything. But if your reaction goes longer, that will be okay. And
This
in
is mine,
fact,
that's yours.
sometimes when your restriction digest doesn't work as expected and
The
you
1x
didn't
TE.
cut everything,
And then, yeah, so we should probably measure this out now.
usually the first troubleshooting you do is to just increase the time. So we'll go to
paper
lunch
man.
and it's okay if our restriction digests end up in the incubator longer than the time it says on the protocol, as long as we hit the minimum time that we know will work. Alright, so we've got our buffer, and we don't have a big enough jar, so I'm gonna actually just dump this extra water out. This mixes pretty easily, so I'm gonna just pour it in here and kind of swirl it around instead of doing the stir plate like we did when we made PBS, because I'm not trying to dissolve powder in this or anything. And then Mike will use this Now, when you're in a lab that's running tons of gels, a microbiology lab, they'll often have a big carboy like this or even bigger, where you can just, it's already made to 1x. And if you're an intern, your job will probably be making sure that that's full and making a whole bunch. So you'll be making like 10 liters of a 1x solution from your 10x. So, all right. So Mike is over here. He's measuring agarose. Agarose is like kind of like a sugar. And when we mix this with our TAE buffer, it's going to turn into a gel, as the name implies, and harden. And that's going to form a whole bunch of little pores within our gels that our DNA has to get through. And because we talked about this in the lecture, the bigger pieces of DNA, it's going to take them longer to like worm their way through those holes. So they'll be where on the gel at the end? The top or bottom? The top, right? And then the little ones can like wiggle through the pores much faster, so they go down to the bottom. And when Mike came up to this balance, what were some of the things he checked to make sure that he could use this balance? It's level, right? Remember we did this last time, so there's a bubble on the back, and he made sure that that bubble was in the middle, so he had an accurate measurement. And then once he put his weigh paper or his weigh bow on the balance there, what button did he hit? Yes, tare, right? To zero it out. And then how much agarose are you? We're making a what percentage gel?
We're making a 2% agarose gel.
A 2% agarose gel. So we, and how much
Yeah.
volume do we need to make our cast?
We need 50 milliliters for the gel.
50 milliliters.
So it's actually in your protocol. I don't know if you all did that calculation yet.
So let's, if you haven't, or let's review. So how much agarose in, so weight by volume, always, it's grams by mils. So the grams need to be 2% of 50 mils. So how much agarose are they measuring out to make a 2% gel?
I have to make one correction in the worksheets that we did in the last session. It was 1% is 1 gram in 1 liter. That's incorrect. It's 1 gram in 100 mLs. Just make that correction and
Yeah.
remember
Okay.
this. It's 1 gram in 100 mLs and not 1 liter. Okay?
Is 1%.
Yeah.
It's 1%.
The easiest,
yeah.
I was saying, the easiest way for me to remember, like Rachel was saying, it's grams over milliliters, so you can make it as an algebra equation, right? So 2% as a decimal is 0.02, right? So my equation to figure out the mass of agarose is 0.02 equals x over 50. Solve for x.
Does that make sense?
That's your equation.
So how much agarose are they measuring out? Not one, no, they're measuring out grams actually. So 2% in, if we're doing 2% in 50 mils, how much agarose is going to be in each gel? And you might need to jot it down on a piece of paper.
So 0.02 times 50 is what? What's 2 times 50? What is it? How many decimals were in 0.02? How many places? So move 100, 2 decimal places.
You had said the wrong units. Yeah, yeah.
1.
Yeah.
1
so
gram.
They're doing 1 gram in 50 mils to make their agarose. And this is going to be a powder. Are we going to stir it up, Mike, like we did with PBS?
No, so
No, we're not.
no.
Yeah.
So the first thing we're gonna do is we're gonna measure out 50 mils of the 1x TAE that Rachel just made, and because we're trying to form a gel, and because of how agarose kind of polymerizes, we actually have to heat this. So we're gonna put it in the microwave for about 45 seconds, and then add more time as needed until it completely dissolves. Okay, at that point it is gonna be very hot, and so just Christy and I are gonna be very careful and only hold at the top of the lid with a paper towel, so it's more insulated, and then we're gonna slowly pour it into the cast, okay? I am going to take this cast to that end of the room. Discrety's gonna stay here, so maybe like half and half, you all can watch what both of them look like, and we'll go over kind of why each component of this matters. Cool? Hang out right here.
So we need
Okay. So this is the general casting. The large rectangle is for a larger gel, and that corresponds to the teeth, right? So large gel, you can fit more bands of plasma, basically. This plastic case is what we're actually going to put into the gel electrophoresis box. The lines, as well as the top band at the top, kind of helps us visualize as the gel is running where our colored bands are going to be. So we put this in first, and we put the teeth in on top. The wide versus the thin doesn't really matter too much, except the thinner one gives you more potential samples. So I'm going to use this one. It has a total of 17 teeth, so 1 ladder lane plus up to 16 samples. Okay, and so I'm going to put that there, and then I'm going to get my TAE, add the buffer. Once it heats and makes a gel, you'll see it's like kind of thicker. We'll pour it in. Before we pour, we're going to add the Saver Safe, and then we'll pour it in, and then we'll wait like till after lunch. We'll be good. Make sense?
What's the cyber safe do?
Say again?
What's the cyber safe?
So that's after we heat it. Oh, what does it do?
Yeah, for that.
Sorry, what is-
I know what it does.
What does Cyber Safe do? Why do we add that? Excellent. Did we all hear that? So whenever we finish running the gel, we're going to put the gel in. We're going to put the gel over a UV boxes in the back and in its UV light, right? And the hidden bromide or the Cyber Safe actually helps us visualize what those bands look like under that light. All right, so this is my 1x TAE. There's 50 mils here, so I'm going to pour this into here. Okay. And then I'm going to take my 1 gram of agarose that I measured out, and I'm going to pour it in.
And this is called a comb, and this helps in making wells in the gel.
Excellent. And again, we were saying we're making a 2% gel today for various procedures. If you're looking for different fragments, it might be longer or shorter. You can alter the percentage of gel. It's just going to take more time or less time for it to run through. And so that varies based on different protocols. And so I might swirl this gently, and then I'm going to
So Mike,
wait for just
or well, for all of you, so if we had a higher percentage of agarose in there,
If we had a
do you
4%
think-
gel,
yeah,
for
do you think
example.
the pores are going to be bigger or smaller? What do you got?
I'm hearing it, but you guys are too quiet.
Be confident.
Yeah,
What were you guys saying? I just heard a bunch of people say it.
you're saying it in a very small voice, but you're right, smaller, yes. Right, because you're gonna have more of those sugars in there, they're gonna cross up and it's gonna be much tighter. So is that gonna be better for separating bigger things from each other or smaller things from each other? Smaller things, good. And so the same thing if we do a smaller, a lower percent, like a 1% or a 0.5% gel, what are our pores gonna be, bigger or smaller? They're gonna be bigger, right? So that's gonna be better. We have bigger things that we wanna be able to separate. So if you have a band that's like, you're expecting to be 5,000 and when you're expecting to be 4,500, you might want a 1% gel or a 0.5% gel so they can separate out from each other and you get more resolution. So you can really see them separate. Or if you did them in a 4% gel, they'd both be stuck at the top and you wouldn't be able to see what it was like. But if you have a band that's like 200 and a band that's 500,
Right after
you're gonna
it's
want
gel.
big pores because you want the 500 to
microliters.
have different difficulties getting to the gel. Does that make sense? Great.
Oh yeah.
Were you
You
guys
have a
able
Cyber
to
Safe
hear
open?
all of that as well? Perfect.
Okay. You can use it first if you want.
All right. So you're heating
Oh yeah, so it's
everything
heating. We only have, actually we can do aliquots while it's heating. Can you make a aliquot for me?
up? On the ladder?
The Cyber Safe.
Oh, yeah,
Cyber
yeah.
Safe, sorry.
Is there only one of them?
Yeah. The tube is like 400 ml, like I just need 50 microliters of it.
And did you guys go over here what the cyber safe does?
No, maybe.
Yeah.
I can unfold it and then.
Yeah, do you guys, so
So-
why are we adding the cyber safe? Do you know over here why we do that? What color is our DNA right now? It's clear, right? So how are we gonna see it once we run this gel? Like how are we gonna be able to visualize it? We need to add something that will stick to that DNA so that we can see it, how we're gonna visualize it. And we're gonna use UV light to visualize it. So the cyber safe will stick to our gel and then it just will look clear when we're staring at it. But then when we stick it on a box a UV box and look at it under UV light, those bands are gonna light up. So, and it depends. Sometimes, like I think you guys have this machine in your lab. Like there's different types.
Oh, the camera?
Yeah, camera. So maybe you'll have to use a fluorescent tag to visualize your DNA. But you need some way to see it, because otherwise you're just gonna be staring at a clear gel and you have no idea what size your bands are if your reaction works.
Other note as well, you'll notice, just Kirthi and I both use containers. Similar to, she had a flask, I had kind of a bottle. Whenever you're doing this, because you have to heat it, it's very important that you use something that is necked at the top, because you're heating it and you don't want it to bubble over. So if you use a beaker where the diameter is the same, it might spill out while it's heating.
Yes. And if you screw a top on your jar, it will explode.
Yes.
Don't do that.
Please don't do that.
So you can put a top very lightly on, but you need to be able to release pressure. And I've seen it many times where people screw and then.
So I had to heat this up twice, but it is now relatively clear. However, it's also very hot. Sometimes you have like hot gloves. Paper towels kind of work okay as far as insulating. And we're going to, you can kind of see now like it looks a little bit thicker. It's not quite too liquidy.
And you're looking for, like, sometimes you'll see some little pieces that look like gel floating in there. And if you see that, you see it just a little more. But that looks clear, but it's a little thick.
Yeah.
So hopefully you guys can see. So it's clear but thick. And if you saw stuff floating in it, which I assume Mike saw after he did the first heating, you just heat it a little bit more.
Yes. All right, so I'm gonna come over here to my group.
Yeah.
And so the last step is to add 50 microliters of the CyberSafe. Right. You were saying before, this is going to help us visualize the DNA actually on the under UV light. I'm going to add this directly into the gel. And then I'm going to gently swirl this for 5 to 10 seconds so it looks relatively uniform. Now, keep in mind, depending on the concentration of the gel, this may or may not be a more time-sensitive experiment, because keep in mind it is a gel and it's at room temperature now, which means it's slowly hardening, right? So I don't want to be too slow in doing this, but I also don't want to rush and risk injuring myself or something, right? But while it's still hot, I'm just going to pour. I'm going to use one location, so I find like pouring into a corner and letting it flow reduces things like blurry lines and other things you don't want in your gel later. So just pick one spot, usually a corner, pour it all the way in. Okay. And I'm going to, again, go fill this with water, let it cool off before I touch it with my hands again. But I'm gonna leave the gel uninterrupted, and in about 30 minutes it should be solidified. It's stable though, so it doesn't have to be immediately used. It'll be good to go anywhere from 30 minutes to 24 hours even. Right? I see for some reason a bubble I don't like.
30 minutes.
So I'm going to slowly drag this to the corner. I'm not going, it's not going to matter as much. But I'd rather not be right in like the middle of a lane or something. Any questions on that procedure? Yes. Yes, to my understanding. I haven't used it before. I used the lithium bromide, but it's supposed to have the exact same function. Yes. Good. Let me get rid of this. And Rachel, let us know what we're running after the circuit's done.
Yeah, I think there are both two questions about lithium bromide.
Yeah.
I would say most labs are phasing out lithium bromide because it's like even the lab, you have to have a special disposal even for it, and that costs money. You can't just throw it in the regular trash or even regular biosafety trash. So I would say, especially as you guys start working in labs, it'll be less and less common to see it. Probably still some in academic labs, but CyberSafe and the less toxic versions are more common.
It gets underneath a little bit, you'll just peel it off.
But if you ever do use it, you'll go through training, and there'll be very strict protocols. We've got our restriction digest sitting, being
Yes.
cut up.
Mhm.
We've got our PCR reaction running through its 14 cycles, and we've got our gels setting up. So you see that's all really nicely timed. When you can do that in your lab work, that's always, you know, you're getting very good at lab work. Because it's lunchtime now. So we just go to lunch, and when we come back, everything's going to be ready for the next step.
Okay. So if you all look on the board, hopefully you remember like what your labels were. If you are designated right now in the braces A, you're going to be doing that here. If you're in braces B, Discography is going to have your PCR restriction digest samples. Okay? So is this only group A people? Yes? Okay. And, okay, so we can start here, so, while we're waiting. So everyone's restriction digest, it should be here. So you're going to be adding 10 microliters of the buffer. There are 2 vials of it here. And you're adding 10 to your sample. So, fine, so I guess that'd be easier. Who's YA one? Perfect. RDAC? I have restrictions, yeah. BD? JW? Satan?
Here, and here's
ADRD?
Satan PCR.
Everybody got one?
YA1 PCR. Great.
So one, make sure you can distinguish your PCR from restriction digest. You have 2 different labels.
I think this is B, so whoever wrote
No, so this one, restriction diagnosis. Our restriction diagnosis is that one, so it's 8. And 10 to PCR.
They're both of them are currently being used, I think. Yes.
10 is to that one. 8 is to
10 goes in the red one, 8 goes in the clear one. 10 in the red, 8 in the clear. Yes, that's good for both. Also, while you're waiting, feel free to take turns coming around. You can touch the gel. It's solidified at this point. Yeah, see what it feels like. You can touch it again once I take it out. But it hardens in about 20 minutes. Once you've added your blue dye to both tubes, just set them down once everybody's done before we actually load the gel. Yeah. Oh, I didn't get the. I forgot the tops. Sorry. Can you come with me really quick? Rachel, we back in 30 seconds. Yes. Okay. Really quick, I just want to see how I should renumber this because I want all the groups to be together. So if you're in a group of 3, you're going to do your restriction digest twice, so everybody gets to inject something. Okay. YA1 and 2. Are you a group of 3 or no? Yes. So I did this earlier. It's fine. Quickly. All right, so lab is going to be lane 1, so you are going to have lanes 2, 3, and 4. So 2 is the PCR, 3 and 4 restriction digest, or pick who does which, but you each could inject once, okay? Satan, it's just 2 of you, right? Okay. Thank you. Excuse me. All right, so you all have all your samples, right? Perfect. So next, everybody get a chance to touch this, just so you can see what it feels like. Everybody touch it. No, that's all right. I'm not gonna bite. Yeah. and you'll actually be able to see once this is done running how thick the gel is. I'm going to leave it in the case again. So I'm going to very carefully pull straight up on the comb so that I don't mess up the lanes that I've created. So it's kind of tight, but just I don't want to do it sideways. Okay. And as you can see now, there's 17 little slots, right? That's where our samples are going to go. Okay. So I'm going to do the first lane, inject the ladder. I'm going to show you how to do it. The primary thing here is to make sure you go straight in slowly and do not go back and forth, because if you puncture your lane, your DNA is not going to run properly. Okay. First thing I want to do is kind of slippery. So underneath, some of the gel when I poured it rolled under, and so I'm just going to kind of peel this on the bottom so it's not slipping everywhere. Not a requirement, but if you leave it on, it could slip around once you're actually injecting it, or once you actually place it in, which would not be good. So I'm just going to remove this extra gel from the bottom. Just like this. Okay. This is where my voltage originates, and so I'm going to make sure it's flowing this way down the lanes. And so I'm just going to press it in here. I've added 300 milliliters of my TAE buffer that I made, and you can see here everything is very clearly submerged. Okay, that's the goal. So each of you are going to be adding 20 microliters of your samples, correct? Okay, I'm only going to be adding 10 microliters of the DNA ladder because I don't need as much of it. What is this? It should not be the same. So, or this high. Oh, this is tight for some reason. Okay. So I'm going to set this to 10. Alright, so I've set this to 10. This is the broken one, that's why. No, it's fine. Yeah, it's all good.
There's 2 broken ones.
Clearly. So I thought it wasn't turning correctly either. Okay. All right, so this is my ladder. All right, can everybody, if you can't see very well, please come here. So I'm going to basically be injecting here. If you are on either side, you will be able to see as it goes in, all right? Okay, so 10 microliters, going in the first lane. So I'm going to kind of use my hand if I need to kind of stabilize things, all right? I'm going to insert slowly, make sure I'm actually inside the slot and not outside of it. But I can steady look to make sure it's not like it's actually inside the hole. And I'm going to slowly make sure while I'm submerged. Slowly inject. And then after I'm done, I pull up slowly. Okay. This, I will admit, usually takes some time to get like really good at. The important things are make sure you're straight in either this way, like this is anywhere here is fine. Do not do this, right? Because you're gonna puncture the gel this way. All right. So either way here is fine. Please make sure you are all the way inside the well when you eject. Okay. And once you've ejected all the way, please pull out. All right. If you happen to press it twice, you're going to blow air into it, and that's going to mess up your sample. Okay. So just make sure you're all the way into your ladder, and then very slowly eject it, and then once you're fully ejected, pull out. Okay. So we'll do it one at a time, just so we're not like crowding too much. So YA 1 and 2, who's on the PCR? Okay. So. Make sure you're inside the lane before you eject. Can you feel that you're inside of it? Once you're all the way ejected, slowly pull out. Good job. Okay. Restriction digest, the first one. Okay, way, so we'll discuss it. Once you're in the well, just very slowly eject. Eject all the way before you blow out. Okay. Good.
So we're running the gels now, and it's going to take about 45 minutes or so for those bands to separate enough that we can see them and the gel. So what I wanted to do while we're doing this is give you a real life example of how we would use these experiments and like what the next steps would be. And these are really common things that you would be doing as an intern just starting a lab. So by a show of hands, has anyone ever done cloning or sub-cloning? You have, okay, great. So just one, that's excellent. And does anyone have an idea what the word cloning might mean?
Maybe like making a copy of our DNA.
Making a copy of a DNA, yeah, a DNA, so making like a perfect copy, right? We're just, and the reason why we would clone is because we would want to make a whole bunch of copies of something that we're going to use. Now, Mike touched on this earlier, but I'm going to give an example of something that I would do in the lab. So first of all, I've drawn up two things up here. They're both labeled with the lowercase p, and you'll notice our DNA template was also labeled with the lowercase p. Does anyone have an idea what the P stands for? Yes. A plasmid. Has everyone heard that term maybe before or not? What is a plasmid? Someone else. Someone from the back that's fine. Okay, we'll take the middle. A circular strand of DNA, exactly. So I've got my PAAV-GFP and my PLVX-F-Luke. So these are plasmids that would be really common in my lab. I work with viruses. So this plasmid is a circular piece of DNA, and within it, I have the genome that would go into an adeno-associated virus, and this is the virus I mostly work with. This plasmid is a map of a lentivirus. A lentivirus works a lot like HIV virus. It's a retrovirus. And so it would have in it the DNA, the directions to make the RNA that's going to go into the lentivirus. And then there's some other important elements on that plasmid. And plasmids are really useful ways for us to store little pieces of DNA so that we can make a whole bunch of new copies, so we don't have to keep every time making new DNA and building it up nucleotide by nucleotide. I can just store my GFP, my F-Luke, I can put human genes into here, I can take genes from mice, cut them out with a PCR reaction and store them in one of these, and then I can grow them up whenever I need them again, bacteria, and I just stick them in the freezer when I don't need these plasmids. So there's, I've got the blue and the purple. These are, like, part of the plasmid is what we call a backbone, would have important things that plasmid needs to work when I grow it in bacteria. And the most important element, arguably, would be some type of antibiotic resistance, because that's how I'm going to figure out which bacteria later has my plasmid and not. So has anyone ever heard of ampicillin? That's a common antibiotic, or hygromycin. Those are both common antibiotics. So in our plasmid, we put genes for that antibiotic, and I will explain more a little bit later. I also have something in orange on both of these called a promoter. Does anyone have an idea what a promoter is? Somebody that hasn't answered me yet. And you can take a guess. It's okay if it's I'll give you a big clue. It's pretty much in the word. What would a promoter do? Yeah?
Maybe shows itself?
Kind of it shows itself. It shows itself to the elements that are going to make my gene. So it promotes the activity of the gene that's following it. So it's an element that you need to regulate your gene so that my polymerase knows, like my primer would be somewhere in this promoter, my polymerase would know where to start to make a gene. Because I don't want to copy, there's a lot of stuff on here that I don't want. I just want my gene and my protein at the end. So the promoter is really important for that. And the promoter has another really important property in genetic engineering and molecular biology. Certain promoters work in certain cells. So all of our cells have the same DNA in them, right? But the DNA that makes your heart cell a heart cell, like different things are switched on. and what makes your lung cell a lung cell, your brain cell behave like a brain cell. So there's different promoters that help regulate when a gene gets expressed and what gene's actually getting expressed. So a way you can control, like if I want this to grow really well in E. coli bacteria, I can make sure that I put a promoter in here that's going to work really well in E. coli bacteria. If I want it to grow really well in a mouse liver cell that I have on a culture plate, then I will put a promoter in there that works really well in a mouse liver cell. Does that make sense? Yes.
So can a penicillin works in the same way as
Yep, you can have a penicillin resistance. Absolutely. So there's a whole bunch of different antibiotics that you can put in there to work in the same way. So the other important thing that we have in this, I made a whole bunch of brown lines, but when you design these plasmids, these are our restriction sites. You might have 100 restriction sites in there. Some of those restriction sites will make 1 cut, some of them will make 5 cuts, it depends. And we used a restriction enzyme earlier. Does anyone remember what it was? It's on your notes, maybe. Anyone have their protocol handy? Huh? Yes? BGL1 was the restriction enzyme we made. So that made, does anyone know how many cuts in our DNA? It made 2 cuts, right. So sometimes, so usually these are designed to be modular. So you'll put a bunch of different restriction sites around your promoter and a bunch around your gene, so you can swap these in and out. So something that's really common is to use, we talked about GFP earlier. Do you remember? We can all say it, what this stands for? Do you guys not remember anything from this morning? Say it, please. Yeah, good. From the back, I want to hear it. It begins with a G, its color. Green. Fluorescent protein. Good. Can the back please say that for me? Green. Thank you very much. Thank you. GFP is a common reporter. So if I want to know where my virus goes or where it's working, I'll use a GFP virus first. So like we're using CyberSafe to see our DNA in the gel. If I'm going to inject my virus in the mouse and I want to know if it goes to the brain or the lungs or the liver, I'm going to use a virus that when it infects that organ, it turns that organ green with green fluorescent protein. But sometimes I have another way I can look at my viruses with something called FLUC, which stands for firefly luciferase. It's the stuff that makes fireflies glow. So I have a gene for that too, and I have a special camera in my lab that without sacrificing a mouse, I can inject it with a virus, and while it's still alive, I can put it to sleep, and I can take a picture of it, and I can see where that firefly luciferase is, where it's glowing like a firebug. So what I want to do is I'm going to actually cut the GFP out of here, and I'm going to put the firefly luciferase gene into my AAV backbone so I can make the virus. So that is a type of subcloning. So what's going to be the first thing that we did today that I'm going to have to do in order to do this? What am I going to do? Yeah? I need to cut it out. Does anyone remember do you remember what the cutting it out is called? We use a restriction enzyme. And do we know what that reaction's called? Ligase. Not ligase, but we'll get to that, so remember that answer. What happens when you cut up all your food in your stomach? What's that process called? Digestion. Right, so we're going to digest these plasmids with our restriction enzymes. So first, we digest. And you don't have to, but it's always easier when you can use the same restriction enzymes on your 2 separate plasmids. So I'm going to use one that's very common called XBA1 and another one called NOT1. And I double checked in my sequences and I made sure that there's only one cut side of each of those. So we will do a reaction much like you did. So we only put in one enzyme today, but you can put in multiple enzymes. And I'll have 2 tubes, one with my AAV plasmid and one with my lenti plasmid. And then what am I going to do once I run that digestion? We're doing it right now. I'm am I going to do with that once I've digested these with my restricted enzymes? What's happening right now while I'm talking to you? Yes? I will make sure they don't restill, but what experiment am I going to do? And I heard someone say run a gel, right? I'm going to run it on a gel. So we're doing that right now. So I'm going to run these on a gel. And what's going to be in my first lane? My ladder, right? So I can make sure I've got the right pieces. And then I'm going to run my AAV in my second lane. How many pieces of DNA am I going to have? How many bands am I going to see on my AAV if I use XPA1 and not 1 to digest my PAAV? 2, right? And so I'm going to have a big one. Which part's going to be the big one? The part with the GFP or the rest of it? The rest of it, is it going to be high or low? Exactly. Great. So I'm going to have a high one, and my GFP is going to be lower because it's smaller. And then I'm going to run my lenti on the lane next to it. And how many pieces am I going to have if I digest it with my 2 restriction enzymes? 2. And is the luciferase going to be the small band or the big band? It's going to be the small band. And we'll say it's a little bigger than GFP, so we'll end up here. And then my backbone is going to be, or the rest of it is going to be my bigger band, right? So my gel is going to end up looking something like this. So now, that's cool. I have the parts I want. How am I going to make this construct, though? They're stuck here in the gel. What do you think I'm going to do? Am I going to eat the gel? No. What might I do? I'm going to have to somehow get these out, right? Cut them out. Simple enough. So I want to end up with luciferase in this AAV plasmid. So in this first column, which band am I going to cut out? The top one or the bottom one? Why would I cut out the bottom? So this is the GFP. What am I going to do once I cut that out? Because I don't want GFP, right? I am actually throwing out the GFP. So I'm actually not going to cut out the GFP. I want to cut out this band because this is what I want to use to make my thing. So all of my DNA is right here. And actually a really cool property of the gel is you can dissolve it and purify out your DNA. So I've actually collected all the DNA I'm going to need right in this band. So we're going to cut out actually this top band. And then what about in my second one? I'm going to cut out the top or the bottom bands to make the rest of my construct. The bottom, right? Because I want to put this one into this one. Perfect. So we'll cut the bottom. So this bottom one is called our insert. And then this bigger one is called the backbone. So we're going to cut out these 2 parts, and then we're going to do something called gel purification. And these are actually really simple kits, and it's something you might very well do in your internships. So we'll put these each in a separate tube, we dissolve the gel, and then we purify it. And when we purify it, we use special buffers, and we run it through a thing called a column. Has anyone ever used a column to separate things or purify things in like chemistry or? No? Okay. So it's really common. You'll see these things. They're essentially, they look kind of like this, and they sit in a bigger Eppendorf tube. And inside here is filled with a bunch of material that when you have the right pH, your DNA will stick to it, and then you spin it really fast and you force all the liquids through that column, and the DNA is going to stick to your column, and all the junk gets washed out. And then after you've washed through this several times and cleaned it, at the very end, you'll put a special salt buffer into that that's going to release all your DNA, and your DNA just ends up as a purified liquid at the bottom, usually around 30 microliters. And you'll have a whole bunch of DNA in there. So now, We've got two parts, right? We've got our backbone, whoops, and one tube, and then we've got our insert in another tube. So now what am I going to do? I have to put these together, and what's that called? Ligation, right? So we're going to use ligase. So ligation is to stick something together. So we're almost through our steps here. So number 4, we're going to ligate it. All right, and so this, we're going to use more, like again, we're going to basically put a whole bunch of invisible drops into a tube to make that happen. Yes?
How are the two backbones different? Like, is the only difference that the two inserts are different?
So these backbones can be really different actually. So this particular, so if it was an AAV backbone, it would be smaller, and I would have in this elements that are important for an AAV to work, and this one would have a linty. So there's different regulatory elements that are special to that virus. That's a great question though. Like this backbone would actually be bigger because all the linty elements are bigger. So you can have different size backbones. It doesn't matter, and this is the key to using the same restriction enzymes that makes it really helpful. So that's a perfect lead-in. So I don't remember exactly how they cut, but the plasmid's actually a double-stranded DNA, right? So here's my backbone, and let's, did you guys discuss restriction enzymes and sticky ends? Yeah? Great. So if I use my XBA one up here, let's say, and DNA goes what direction? There's a 5' and 3' end. Yes?
5 or 3, but I have a question.
Yes?
You were saying that you choose the same restriction enzyme?
You don't, there's ways to do it if you don't use the same restriction enzyme, but I'm going to explain why it's best if you digest both of these with the same enzyme right now.
I mean, I get it. So like they could reattach, but like, what if they like reattach in the wrong place? 'Cause you
We'll explain how we're going to screen for that too. That's a great question. All these things you're thinking about are excellent. They're things that people have had to design around. So let's say our outside strand here, we're going, this is the 5' end right here, say, 5', and we're going this direction, so this would be the 3' end. And then what's this end, 5' or 3'? 5'. Exactly, and this is our 3'. And what happens with most of these restriction enzymes is they leave a sticky end. So we have a couple of bases here that aren't attached to anything yet. We have a couple of bases here. And because we use the same restriction enzymes, what's going to happen? Our luciferase, this is going to be 5 or 3' end? 5, right? Because 5 pairs with 3. So our luciferase is going to have the opposite sticky ends. And this is going to be my 5' and my 3'. And it's also going to have the overhang of a couple bases. And those bases are going to be, begins with a C. They're going to be what to the sticky ends have, the bases are what to each other? Complementary, exactly. So what that means is those bonds for a complementary pairing are much stronger than other things going on in a reaction. So when we put these together, the chances statistically of it sticking the right way are higher, but if it doesn't happen in the right way, usually it won't grow correctly, or they won't pair. And then there's something called ligase in there. Ligase essentially goes in and forms the last little bonds, so that we have a complete plasmid now with our luciferase and our AAV. Does that make sense? So, and just for like good review, what does G pair with?
With C.
Exactly. And what does A pair with?
C.
Great. Do you know how many bonds form between a G and a C? Anyone? 3? Yeah, I heard several people say great. So 3 bonds. And AT? 2. So what's a stronger pair?
GC.
Yeah, GC, right. So that's good to know too when we were doing the PCR. When you design primers, if an area is very GC rich, you're gonna have to get a lot more energy in there to break all the bonds. You're gonna have to get it hotter, so they usually have to, those primers have to go at higher temperatures. So it's just, all those rules come into play. So now we've got a plasmid. So we have a little tube of clear liquid, maybe like 10 microliters. That's probably not enough to like make a virus with and use in a mouse, right? So how do you think I'm gonna grow this up, or it's called amplify it? Any ideas? Yeah? All right, so PCR could work, but PCR is better for amplifying shorter pieces of DNA. This might be 6,000 base pairs long, and so it'd be very hard to find a polymerase that would build a template for that long a piece of DNA. But there's something in the lab and in life, an organism that grows super fast, and it copies DNA really well, and it doubles really, really fast. Does anyone know what that is? I probably have a lot of it inside of me right now. Bacteria, exactly. So we're gonna put this in a bacteria. The most common bacteria for this is, does anyone know? Yeah.
Is it RNAs?
Not RNase. So RNase is an enzyme that chops up RNA, integrates RNA. So anything that ends in an ACE is an enzyme, and it's usually chopping up whatever comes before it. So polymerase chops up proteins. Yes? E. coli is the common bacteria used in labs. It grows overnight. You can get a huge colony. So we're going to amplify our colonies. So actually this process of infecting our bacteria with our plasmid is called transformation. So bacteria, and this is part of their survival in a way, they're really good at taking in foreign DNA. And then once that foreign DNA is in the bacteria, it just sits there as a plasmid, as a little circle, and every time the bacteria divides, that little circle gets copied. So then we start with one bacteria, then we have 2 bacteria, 2 plasmids, and then we have 4, and then we have 8, and over E. coli, I think it's every 15 to 30 minutes it divides. So you can imagine if you let that sit for 10 hours, you're going to have a lot of stinky bacteria in a tube. We had the question, how do we know that this works? Because maybe like our ends just stuck together. So we're going to actually select colonies first. So what we're going to do is we're going to, here's our bacteria. We need to, the bacteria has like a cell wall around it, right? So we need to shock it open so it will let in our DNA. So there's a couple different ways you can do it. One's with electricity. The way I commonly do it is called heat shock. So I start with my bacteria on ice. I add in my plasmid. I then put it at 42 degrees for 2 minutes. And that heat allows a bunch of pores to open up in the bacteria. And then I'll end up with my plasmid and some of my bacteria. Some of them won't get it. And then I put it back on ice, and it will close back up again. And then I let it grow overnight. Yes?
So you're saying for 2 minutes. Why specifically 2 minutes? Because I know people do different times.
Yeah, so it would depend on the strain of the bacteria. And I would say it's pretty robust. Sometimes it's as fast as a minute. So it's like the lab. But basically, you don't need to do it for that long. Have you done a different protocol?
Yeah, I did it, and actually I did it 2 weeks ago, and you did it for 50 seconds.
For 50 seconds. So it's probably a better strain of bacteria than the one I use. That it responds to the heat much faster and takes in DNA much faster. So, and that's another great thing, like these are all, you never want to memorize necessarily the times and the exact, so you want to memorize the concepts of these protocols. So the idea of like heat shocking a bacteria so you can get DNA, you want to understand that. But then in your lab, it depends on the bacteria you're using and the equipment you have. So it might be slightly different temperature or time, but it's all the same concept. So then I'm gonna let this bacteria recover for about 30 minutes. And then not all of my bacteria is gonna have gotten my plasmid. So I'm going to spread my bacteria out. So let's make a plate. So here's my plate. And I'm basically, I want to take this, have you guys in your biology classes spread bacteria before? On a plate, streak to plate? Yeah, that's a pretty common. So I'm going to just take, I have a tube of bacteria that I heat shocked, and I'm just going to spread it over this plate. And there's going to be a really important thing that my plate needs to have, so I make sure tomorrow morning that the bacteria I see have my plasmid. Does anyone know what that is?
Nutrient?
A what?
The agar nutrient?
They'll have agar, which will make them grow, exactly. But there's something else, because I don't want all my bacteria to grow. I only want the bacteria that has my DNA in it. Yes. The antibiotic, right. So remember how my plasmid had this ampicillin resistance? So if this worked, and my plasmid's in my bacteria, that bacteria is now resistant to ampicillin. So if my plate is full of ampicillin, then tomorrow morning when I look at that plate, it will look like some white dots. I'll have some colonies of bacteria that are resistant to ampicillin. And that's a really good indication that that bacteria is carrying my plasmid. And because of the way bacteria grow, and because I spread them out, this will be many, many bacteria in this tiny dot. But I will take a pipette tip, and I'll just pick, they're all exact clones of each other. They're just duplicated and duplicated. So I will pick off that clone, I'll stick it in some LB broth, and it'll grow up again overnight. And then I end up with a big tube. So number 6 is going to be, I'm going to amplify my colonies. 6. So I'm going to pick, because this isn't perfect. So we had the question, how do I know? How did I know it didn't go in backwards? Or maybe, because if like these ends just stuck to each other for some reason, I would still have ampicillin resistance, but there'd be no luciferase in there. So how am I going to know? So I'm going to use basically statistics, and there'll be a certain percentage of chances, depending on how many of these colonies I have, that at least one of them will be right. And I only need one of them to be right. So I will pick off 2 or 3 colonies, or maybe if I'm really not sure and I think that there's a high chance that this didn't work, I might pick 14 colonies, and I'm going to grow them all up, and then I'll have a bunch of tubes of stinky bacteria in the morning, and then I'm going to have to purify those. And this was actually the first thing I ever really did when I first did my first lab internship in a molecular biology lab. You're going to do something called a mini prep or a maxi prep. So the next morning, we want to get our plasmid out. So what are some steps we're going to do that? It's in our bacteria right now. So how are we going to get our DNA out of our bacteria? Does anyone remember that word? Yeah?
So, did you use another restriction?
I can't quite hear
Did
you.
you use another restriction?
Another,
To put out the restriction?
not a restriction enzyme. So I want to actually, it's inside my bacteria. So there's something called lysine. So lysine is going to bust open all my bacteria, and I'm going to end up with all the proteins from the cell floating around, and then I'm going to end up with all everything that was inside the cell. And then I'm going to spin it really fast, and I'm going to end up with all the cell wall and the cell components floating to the bottom, and then I'm going to have this, it's called supernatant, or soup. I'm going to have the soup on top, and that's going to have my DNA in it. So then how am I going to get my DNA out of the soup? We already did it once in this process. I'm going to run it on a column, right? So I'm going to push the soup through a column. The DNA is going to stick to the column, and then I'm going to wash the column a whole bunch of times. And then at the very end, when I think it's really clean, I'm going to release the DNA off my column. And again, I will end up with a tube, an Eppendorf tube. And at the bottom of it, I'll do it in pink, is going to be about 30 microliters of my purified plasmid. And now I need to do one more step because I don't know if it's the right one, right? So the very last step would be to QC this, quality control it. There's a couple ways I can do that. So let's say I have 4 of these tubes, and I want to know if 1, 2, 3, or 4 is right. So does anyone have an idea of some of the QC I might do? One QC is I want to make sure there's like clean DNA in there. So there's a thing that you'll see in a lab called a nano drop, and I'll take 1 microliter of that plasmid out and put it on a special machine, and it will read that drop and tell me, it'll give me a number. Always forget this, maybe Mike can help me. It's the A260 over 280, is that the way it is?
There's a 260 over 230 and 260 over 280.
So the 260 over 280 is going to tell me I want this number called a 260 over 280 to be between 1.8 and 2. And if it's too high or too low, it will mean I have a bunch of RNA contaminants or a bunch of DNA contaminants in it. And then the other thing I can do, which Mike talked about, is sequence this. So I can take a little bit of this plasmid and sequence it, and we did a reaction today that's like a sequencing that you can do for sequencing. What was that? It's not a restriction digest, but PCR. So the PCR, what will I put into my PCR reaction for sequencing? You can say louder. I just want to, but I appreciate the enthusiasm. So on your protocol, what types of things did we put in that tube today? We put non-restriction enzymes. We're doing PCR. What are some things? We had a forward what? Primer. We had a reverse. Great. And what else did we have? What's our template in this case? It's going to be the PAAVF loop, right? And we're gonna basically, do I have to sequence this entire thing? No, I don't have to. Because it would be very unusual for one of these parts to have like, they should be correct. So I mostly, where you get mistakes are where these are joined together. So I'm gonna design my primers. I'll make those in brown. So like I'll design a primer that starts a little bit before, this is called a homology arm. So that will be my forward primer. And I'll design a primer that's going to be a little bit after the second arm. And so then what's going to happen is I'll put the plasmid in, and then over the many cycles I'll amplify just this insert with the parts where there might be mistakes. And then I can take that product and sequence it. And the sequencing will give me just a list. It will say something like A, T, G, 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 A, C, A, C. And I will, there's computer programs, so you don't have to sit there by hand. And I'll line them up. And if they line up, I know it worked. And say it lined up for number 1. So I will take number 1, and that's what I'll use to make my virus. Or make even more, it's sort of a bigger stock. Yeah. So this is like kind of like a whole cloning project, and it seems like it's so many steps and it takes so long that you will be amazed because it's a really common thing that interns end up doing. And in like a couple of days, you can have like a whole new piece of DNA. Yes?
Would you blast it?
You can blast it, so that's a way to line up, absolutely. So, but what I wanted to say is pretty incredible about all of this. Does anyone know what year the structure of DNA was discovered? Or have a ballpark idea? 1953. So how many of you know someone that's 66 years old or older? Grandparent maybe. Right, so in their lifetime, like that DNA, like they were babies, DNA we didn't even know what it looked like. And does anyone know when restriction enzymes were discovered, or have a guess? Yeah, in the early 70s, in like 1970. Does anyone know somebody that's 49 years old? Yeah, like maybe a lot of parents or aunts and uncles. So, like in their lifetime, that was discovered. And then how about sequencing the human genome? Does anyone know when those first sequences came out? The late 90s. So 2001, the preliminary sequences were released. So does anyone know someone over 18? Yeah. You know, at least, yeah. some of us in this room are over 18. So like in your lifetime, we know the human genome sequence. And so we've gone in just, from maybe your grandparents' lifetime to yours. Like your grandparents were maybe learned about DNA in high school. Your parents maybe. And now you guys, just on a Thursday afternoon, have used restriction enzymes and cut up DNA and run it on a gel and have all the skills to just make a new piece of DNA for some application. So I think that's really incredible because I'm sure you guys hear all the time like, oh, when I was a kid we didn't have cell phones. So that technology has blown up, but the DNA and the genetic technology, like I would argue is maybe even more, because we're to the point now where we can actually edit genes in human beings, and those trials are starting, or we can add genes to human beings to get rid of a disease or to improve a disease. That's pretty incredible considering, 60 years ago, it would have just been a handful of scientists that actually even knew what DNA looked like and what it might do. So, I think I saw Mike looking at it. Are gels close?
3 minutes.
Perfect. So are there any questions about these ideas or these concepts we went over today? So, I will, yes? How do the primers work? Oh, great question. So the primers, in order for your reaction to know, because we are going to copy this whole thing for the PCR, right? So in order for us to know, for the polymerase actually to know where to start, because then we're going to build on our template, right? We're going to match. If there's an A there, we're going to put a T, and if there's a G there, we're going to put a C. The primer is actually, I think they're usually an RNA probe, but it's complementary to a sequence on your template, and then there's elements in it that the polymerase will recognize, so it knows start there. The primer is basically like a tag that says start here. And it works because it's complementary to a sequence in your DNA. And then once that polymerase, that big enzyme attaches, what's going to happen? What was another thing we had in our PCR reaction? The deoxy nucleotide mix. Does anyone know what that is exactly? What a deoxy nucleotide is? That's a single base. So there's 4 of them, right? What are the 4 of them? G, C, A, T. Yes, right. And so it's your base with your sugar backbone and your phosphate. And what's going to happen is the polymerase kind of holds your template, and those bases will float in. And when you have the proper binding, it essentially, this isn't required, but essentially like pinch down and kind of keep it there and move on. And it will just, the polymerase will start at the primer and then just move along. And we're going from 2 directions, right? So we'll just move along and build up our template, and then our primers will fall off. And polymerases, different ones work differently. So some can only do 100 base pairs, some can do 1,000 base pairs. And so all of that goes into the design. Yeah. Are there any other questions? All right. So what we're going to do is, and we'll have to do this in groups, we're going to look at our DNA. If you want to get your phones out, you can take a picture, and then we'll finish up by, I think, do they all have a ladder picture in their packet? Do we have a picture? We just have a couple. I saw some.
I emailed it to the script. It wasn't in the protocol.
Okay, I think we've got a couple that we can set on the table, and we'll figure out what size our different bands are, and if it was right. So, and then what time do you have? Okay, then we'll do a little pub trivia like we did last time, and our original groups from this morning, and then we'll call it.
So it's not as bright as I would like, but can you see where the bands are?
Yeah.
And so some of them are more distinct than others.
Maybe we can go to the class after this and then flash it.
Yeah.
Make sense?
Do you want to see it, Rachel? You can see the bands are they're not super bright. Like you can use like a photo editor to brighten them, but like they're there, they're just not popping. Part of it might be, like I didn't look at the protocol for the CyberSafe, so I don't know if we didn't use the right volume.
Yeah, so it's as though you guys should have to Oh, I don't know. You guys just think you guys usually do 10x, so it's really good to do like Yeah, you should use like a micrometer per mil. Like cameras and machines that you just take a picture of it, and then if I was gonna like show this in a presentation or something where all my bands were, I would just on the computer like pump up the brightness. But you just really need to be able to see it enough
This is a better
to
ladder
like
too.
be able to punch out your DNA.
Group B, you can come look at yours as well. So you don't even know if you actually see it yet, really? You're gonna know if it's right or not. I don't recommend the flash, personally, but
Really? You had more? You had multiple bands in the PCR well?
I think they mean lanes.
Oh, okay. No, so all the lanes that were PCR reactions, how many bands were on that gel in those spots? In the PCR? Where's the second band coming from? There's one, right? So the PCR reaction, we were taking a DNA template and we were amplifying one piece of DNA. So we should have a whole bunch of one piece of DNA. So we would just have one band, right? And what about in the restriction digest lanes? How many bands do we have? 2? Did you guys see 2? And why is that? How many cuts did our enzyme make? 2. Right. What? So if you saw 3 bands, what might have happened? Does anyone have an idea? Problems would have happened. I've had this happen in reactions.
There's one, I don't know if they had it this year, but there's definitely one from, I'll show you on the next slide, that has three bands. Yeah.
Yeah. So sometimes, sometimes, remember we said you have to be careful pipetting because you can mix up. So sometimes you don't have a pure template, so you might have a second template that got in there, so you're getting extra cuts or extra things you don't expect. Sometimes, and this has happened to me, you have a map and you expect the restriction sites in a certain place, and maybe whoever made that map or those sequences made it wrong, made a mistake, or at some point your DNA mutated and there became another restriction site. So you actually got a cut you didn't expect. So I've definitely had that happen where I have 3 bands and I only expect 2. And sometimes that's okay depending on where the cut is. So if I had, let's say, I don't know that, do we need this? So let's say, here's my plasmid, here's my GFP, and let's say actually my restriction enzyme, there was a cut here, here, and in the GFP. So how many bands am I going to end up with here? 3, right? If I want to use the GFP, does it matter? Yeah, right? Because I've cut my GFP in half. But if all I care about is the backbone, does it matter that I have this extra cut in the GFP? No, because I'm going to throw that away anyway. I only want this. So sometimes you might only have certain enzymes and they're making more cuts than you want, but you might get lucky. And if I want the backbone and I have an enzyme that will give me 3 cuts and it's just chopping up what I'm going to throw away anyway, I can still use it. Does that make sense? So yeah, so as you get more into cloning, there's all these other things that tricks.
One other example. So I did this for the first time in a class 3 years ago, and this was my gel, or my group of like 3 or 4 people. So these are both supposed to be a restriction digest, and so I expected, for some reason this lane kind of drifted here, but this I expected to be kind of my higher lane. What do you think this one is at the top?
That's great.
The largest strand.
And how big should the largest strand be?
The part that you're not cutting out, like everything else.
So in our case, we had Yeah, so we basically had this, right? So we had one slightly shorter and one slightly larger, right? But what if I told you this is the longer fragment and this is the shorter one? What's the thing at the top? Hypothetically.
Maybe the plasmid didn't get cut at all.
Yeah,
Yeah.
right? So part
So
of
the whole thing's gonna be even bigger, right?
Mhm.
And do you think a circle moves easier or like faster or slower through pores? Slower, right? Because the circle has more places it can get stuck. Yeah.
right? And so a component of that as well is the number of cycles you run. And so you can actually do the math to track exponentially how the number of cycles, as the number of cycles increase, the number of ideal fragments you get actually increases, but we didn't do a PCR for the restriction digest. So if you have a third band on top that is actually higher than your ladder, right? So this is my 1500 band on the ladder,
Yeah.
right? Remember that ladder I showed you guys in the lecture earlier? So this is the highest on the ladder, and that's higher than that, right? So if we see a third band that high above the ladder, chances are some of the plasmid didn't get cut properly.
Does anyone have an idea of how the next time we can make sure it all gets cut? What's the first thing we could try to do? Yeah?
Actually use restriction enzymes?
Well, no, we used because we've got 2 bands, so some of it got cut. But do you remember what I said before lunch? What happens? What matters for the time?
Increase the time?
Increase the time. So maybe we didn't give our enzyme enough time to cut through everything. So it only had enough time. So the first thing you could do is increase the time. What's the next thing you could do? Maybe we didn't have enough enzyme. We use it all. But these enzymes usually are made so they can cut a lot of DNA, like far more DNA than you would ever put in. So usually if you have not enough cuts, you haven't run your restriction for long enough. So you just increase the time the next time. Yes?
Can you simply increase the number of cycles to increase the number of ideal fragments?
So yeah, that would be for PCR. But for a restriction, if you weren't getting cutting, if everything didn't get cut, you just didn't give your enzyme, usually, you didn't give your enzyme enough time to cut everything. So you just increase the time. Yeah.
So yeah, I don't know, the second image is kind of blurry, but this one you can kind of see, so I can clearly see, right, so I know, I can't remember if this is group A or B, but this looks like a PCR followed by 2 restriction digests. So this might have been group A, so I know I had that in my first set, right? So you can kind of see very clearly the restriction digest, right? These look beautiful, right? You can clearly see both the bands, right? The ladder is a little blurry as well, but you can see as well on the PCR, it looks like we have bands above and below. Y'all see that?
Yeah.
I see it at least. Is
And
it
all of
most
them it
lines
looks
actually?
like, yeah.
On the, yeah. 16 and 14 too.
So what do you think that is?
What do you guys think?
Or do you think there's 2 bands there? Because we only expect 1, right? Because we were amplifying 1 piece of DNA.
So let's start here. Which band do you think we should have, right? If there's 2 we know we should have 1, which of them should we have and why? Do you remember when I drew on the board earlier how long the plasmid we used, that PUC19 vector? Remember how many base pairs I said it was? Say again? Not quite.
2,700.
Yes, right, about 2,700 base pairs long, right? So that should be above my ladder, right? So if anything, I would expect this top line to be my PUC vector. But the difficult thing within PCR is that a lot of it is relative to time, right? And so this is when the number of cycles you run actually kind of matters. So for example, I won't go to the math, but if you ran, for example, let's say you ran 3 cycles, right? So I'm going to show really quickly. Yeah, okay. So there are some like animated videos that might show this a bit better, but let's say this is my DNA, hypothetically, right? Let's assume it just got separated, and I'm going to add a primer here and here, right? Now, you'll see in your procedure the elongation step. We had it for 3 minutes, but various enzymes and plasmids work differently, and so the timing may or may not need to adjust. But what happens if, for example, by the time the cycle ends, we stop here? So now, we have 4 strands of DNA, 2 different sizes. Do you see that? So a lot of how various fragment sizes end up arising is just because of the enzyme elongating for various periods of time and adding nucleic acids. This is why, ideally, as you produce more and more of your cycles, these unnecessarily shorter fragments end up being phased out. Does that make sense? I can send just to you like an animation as well that might articulate it a bit better. But more or less, the fragment you're trying to obtain is often different length than a potential length of the vector.
And yeah, can we discuss too, like, just to remind us the steps that are happening in the cycler? So what's the first step that you do? Yeah. Denaturing. So you have to like melt the DNA so you end up with 2 separate strands, right? So if that wasn't at the right temperature or long enough and your DNA didn't fully separate, maybe the site where your primer has to bind isn't available, so your primer isn't getting it. And so then what's the next step? The annealing. And what's happening there? Yeah?
The primers stick to the DNA.
The primer's sticking to where they're complementary, right? So if that step, like something's off in the timing or the temperature, because different primers will have different annealing temperatures, remember, based on the amount of G's and C's in there. So that, if that temperature, sometimes it happens, you usually try to design your primers so that they work at the same temperature, because if your forward primer works at a higher temperature than your reverse primer, depending on what temperature you set your cycler at, only one of those is going to bind, so you're only going to get half of the fragment. And then what's the last thing? Yes. The extension, right? And so that's what Mike's saying. If that's not long enough, you might end up with fragments that are shorter than you expected. And so then depending on where it might be going wrong, you'll end up amplifying the wrong size band, which is why it's really important to check on a gel and see. And so then, like, you could use this to assess and like, okay, I'm ending up with a band that's too short, and you can figure out what size it is and kind of guess like, okay, this might be coming from the elongation step. So the next time we run that program, we're going to increase that by a minute and see if that helps us get that full DNA. And I think an important point in that is to become a good researcher and a good scientist. I did say it's like cooking, but you're not blindly following recipes, right? You want to become a chef. So you want to be able to bring in ingredients and like taste it and figure out if there's too much salt. And so the better you get, you don't just look at this data and be like, okay, and you show your mentor. I mean, initially you're just like, yeah, this is what happened, I don't know. But you want to always be thinking like, wow, that's not what I expected. Why do I have 2 bands and what might have gone wrong? And each time you kind of learn tricks and what you should look for next, and you get better and better at troubleshooting. And then eventually you discover something and you change the world, maybe. But yeah, so it's always important to like take your data and your experiments and assess it. You're not just, maybe you want to be hands, but I don't think you're all here and going into these internships because you just want to be somebody's hands. You want to be a brain too. So, all right, is there anything else? Great job, you guys, today on all of this. And so we're going to finish, we're actually going to do the individually today, our trivia. And I don't want you to think of this like, this isn't graded or anything. This is really to help us understand what we have and haven't communicated properly to you today. So I know it's the end, but we are getting out early, so you can go enjoy the weather. So we have 10 questions and a bonus. So I'll go through them now and make sure your name's on your paper. And and we'll film it as we
They
go.
can
So
go
my
back up,
first
yeah.
question
I mean, I
is
think
what
so.
is the name of the piece of equipment that we ran our PCR reactions on? So just keep it to yourself, just write it on your paper. This is individual. Does everyone have a piece of paper? PCR equipment. Sure. All right, number 2. What was the percentage of the agarose gel that we ran today? Or made and then ran our samples on? What percentage of gel did we run? Just keep it to yourself, and this just helps us assess how we're communicating. Number 3 has 4 parts. Part A, what charge does DNA have? Part B, what part of the DNA gives it that charge? And it's okay if you don't know. We'll go over them at the end. Part C, huh? Yeah, I know that's what it's on there. Part C, what direction is the current running in our gel? What's the polarity from what to what? What charge to what charge? And then part D, when loading our gel, the wells should be oriented towards which end, which charge of the gel box? Number 4, if you're trying to separate larger band sizes, do you need to increase or decrease your percentage of agarose? So when you're trying to separate large band sizes, do you need an increase or decrease in your agarose percentage? Number 5, what product was used today to visualize the DNA in the gel? Number 6 is a real trivia. What year was the structure of DNA discovered? For all the Jeopardy dollars. Alright, number 7. What is the purpose of the primers in the PCR reaction? Number 7, what is the purpose of the primers in the PCR reaction? Number 8, what is in the deoxynucleotide mix? What is in the deoxynucleotide mix is number 8. Number 9, get ready, Mike. I'm going to give you a sequence of a forward primer, and you're going to tell me what the DNA it will bind to is, what the DNA sequence will bind to. A, T, C, G, A, G, C, C. So my forward primer is A, T, C, G, A, G, C, C. And you're going to tell me what DNA sequence it will bind to. Number 10, you're going to tell me, this is part A, which 2 bases form a triple bond? Part A, which 2 bases form a triple bond? Part B is what 2 bases form a double bond? Part C, which base pair takes more energy to break apart? And part D, in a PCR action, what supplies the energy to break those bonds apart? In a PCR reaction, what supplies the energy to break the bonds of our bases apart? And then the last part is somewhere on your page. Number 11 is just a bonus, and there's many answers that will work. I want you to give me a reason why you might want to amplify some DNA. Like what might be an application of that, or a piece of DNA, and can we say how you would do it if you want. But why would we amplify DNA? There's a lot of different answers that can be. So we'll give you, does anybody need any of those questions repeated? Number 8? Number 8 was...
What is in the deoxy nucleotide mix?
Yeah. So we use that in our PCR reaction. I want to know what was in the deoxy nucleotide mix. So we'll give you another minute or so, and then if everyone can just pass their pages forward, and we'll go over them so you don't go in out. What was the equipment that we ran the PCR reaction on? Thermal cycler, yeah, thermal cycler, good.
Do you guys understand why? Why is it called a thermal cycler?
Yeah.
Thermal
Yeah,
and
heat.
cycle.
And it goes through many cycles, right? Cycles through, right.
Engineers are naming things.
Engineers do name things very well. What percent gel did we run today? 2%, right. What charge does DNA have?
Negative.
Excellent. And what gives it that charge? Do we remember? Phosphate. Excellent. What direction does the current run in our gel box?
Negative to positive.
Negative to positive, perfect. So when loading the gels, where do we need to put our wells? Towards what end? The negative end, right? So we repel, push our stuff to the positive. If you're trying to separate larger size bands, do you want to increase or decrease your agarose? No, you actually want to increase it. Right? Yeah. So let's think about why. So if we want, is that right?
No.
So,
Sorry.
Wait a minute.
increase it to try to separate larger bands.
So we have larger bands, so we want them to. No, maybe I said it backwards actually.
No, it's lower.
Yeah,
Because-
decrease. Yeah, sorry. We were all right. It's also definitely So our pore size is going to be bigger or smaller if we. It's going to be bigger, right? So then our big piece of DNA will be able to get through a little bit faster, and we'll get better separation up high.
So if we made a 1% gel today instead of a 2%, those same bands that we found would have drifted further in the same amount of time, right?
Where the opposite, if we have really small bands we're trying to separate, we want to increase it because we want the really small stuff to have a harder time. Like we want small stuff to have a harder time so we get a bigger separation. And that's why the ladder is so important, because that ladder will move around depending on how big your pore size is. Yeah, great you guys, excellent. What product did we use to visualize the DNA in our gel? Right, so I realized after the fact that might be. So what was the one we put in the gel that would stick to DNA? Cyber safe instead of iterative growing, right. But I understand why you use the blue juice, because to see our reaction running down the lanes, we use the blue juice. So that works too. But the cyber safe is the key part, because could you see the DNA? If we only have blue juice in there and no cyber safe, when we put it on the UV, would you have been able to see the DNA? And where's our blue juice at the end? It's at the bottom, right? Because it runs faster than a reaction. So all the blue juice is a line at the bottom. Has nothing to do with where the DNA actually is. So to actually see the DNA in the gel is cyber safe. Or we didn't use this, but it didn't bromide. Great. So number 7 was, what was the purpose of the primers in the PCR reaction? You want to give it a try? No, I think you were saying it. To make a copy of DNA close, what do they, yeah, you want to try?
Stick to the DNA.
To stick to the DNA, and what's the point of it sticking to the DNA? We're gonna get it all together.
Yeah, close.
We're getting close.
Does it recruit the polymerase?
Say it louder.
Does it like bring in the polymerase?
It brings in the polymerase, great. And what's the polymerase do?
Doesn't it like tell you where to start the duplicating?
Yeah, exactly. So the primer is like our start point, and it brings our polymerase in, which is what we need to add all the nucleotides. So if we don't have a primer, the polymerase doesn't know where to go, and it won't actually start building template on our DNA. Yes?
So what if you said like it contributes to the directionality of DNA base pairing?
It does contribute to the directionality. Yeah, that is
What does directionality of DNA base
Yeah.
pairing mean?
It means like okay, DNA polymerase is going one way, so it tells you which way to go, so you don't go backwards.
yeah.
Isn't DNA double-stranded though?
Yeah.
So aren't you going one way on one strand, the other way on the other strand?
Yeah, but what if that's reversed? Aren't you going the wrong way?
So the polymerase
I'll stop giving out retarded.
the polymerase can only build, like the nucleotides can only come in one direction because of the phosphate actually and how it interacts.
Yeah.
Yeah.
Like, have you heard 5' to 3'?
Yeah.
So nucleotides can only be in a certain order anyway. So the primer doesn't tell directionality.
Yeah.
It just tells the location of starting.
Yeah, the primer tells you where to start. And if you said something about it needing to be there for the polymerase to know where to go, which is activate the polymerase, that works too. What year was the structure of DNA discovered? I skipped that one. 1953. Yeah, it's okay. Yeah. But it's just, the bigger thing is just to realize it's really not that long ago when you think about it, that like we had no idea what DNA even was. So.
You don't know who discovered it?
And there's one more person.
Somebody said it actually.
It's a her.
It's a hers. I heard it down here somewhere.
Yes, Rosalind Franklin.
Yes.
The person she worked with in her lab, though. I forgot his name.
Oh, yeah, I don't know, but I always forget which one stole it, basically.
Yeah.
Yeah,
I think
yeah. So Rosalind Franklin, and she's just now getting a lot of credit.
Yeah.
She was, one of them was working on a dry form, and one of them was working on wet form crystallization. And it was actually her images that allowed them to see that it was a crystal structure. And she actually died before the Nobel Prize was given out. So, but yes. All right. Where am I at? Primers. What is in the deoxy nucleotide mix?
DNA nucleotide.
Yeah, we're kind of getting there. So I heard sugar. The sugar's attached to
Technically
what?
partially.
Yeah. Yeah.
Single nucleotide base.
Single nucleotide bases, right? So that includes your A, T, C, and G, your sugar backbone, and your phosphate. So they just load in one at a time. Mike's gonna draw it up. Yeah. So you have to have those in there, because otherwise you can't build a template, right? You need A, T, G's, and C's. Yeah, perfect.
So wait, is the answer all three of those or just the bases?
This entire thing is a nucleotide.
That is a nuclear. So if you said like individual bases, or you described like a base with a sugar backbone and a phosphate, or but the idea we want you to get is that you have to have A, C, T's, and G's floating around to be added to your DNA template. Otherwise, you're never going to build a piece of DNA, right?
But if you just said phosphate groups, that's not technically
No,
true.
I just said
Oh.
ACTG.
Yeah, that's fine. Yeah, but and what we want you to understand is it's the whole part, and then that's going to be built onto your DNA.
Yeah.
Great. So my forward primer. We'll do this one at a time. What's going to be the DNA sequence? So I start with an A. What's wrong with my DNA? T.
A.
C. G.
C.
A.
T.
G.
C.
C. Excellent. Number 10. Which two bases form the triple bond? G and C, right. And which forms the double bond? Great. So which pair takes more energy to break?
G and C.
Great. And in the PCR reaction, what supplies that energy? Heat. Yeah, exactly. Heat. And then for a bonus, we'll just take a couple people if you want to give share your answers. A reason why you might want to amplify DNA. Yeah.
To create a larger sample size to lower your margin of error.
Okay. Great.
Maybe to take different samples and put in different like animals.
Okay, so you need to make up enough DNA that you could actually put it into an animal or a person for a test or treatment. Great. There's some other reasons why we might want to amplify DNA. To make DNA what? To remove a disease? Right, so that wouldn't be like making, so the idea of amplification is taking a small amount of DNA and making a large amount of it. But if you were making a large amount of that DNA so that you can study it and be able to use it in your experiments, definitely. Great. Is there any other reasons we can think of? Yeah?
I also want to limit the amount of maybe a specific gene that's DNA that you want to put that could produce a certain molecular compound like DNA, so that you can increase that amount, I guess, like also increase its availability to people and also mitigate the price of it.
So kind of like increasing the amount of DNA, of a specific piece of DNA that you could then use in a treatment. So something like insulin, great. Does anyone think of any other reasons?
I was saying fingerprinting.
Fingerprinting? Yeah. So if you add a little tiny DNA sample at a crime scene, like a little drop of blood, you could take and amplify that DNA so that you have enough of it that you could look for a signature, like a DNA fingerprint, and figure out who committed the crime. What about something like 23andMe? Do you think they amplify your DNA? Because you're just spitting in a tube. Do you think they can get all that information from that little bit of spit? So they have to amplify your DNA. Yep.
Did Elizabeth Holmes, the woman who discovered the out of blood test?
Oh yeah.
Did she do the same thing for people?
She was not doing what she said she was doing. Yeah, she wasn't amplifying. The idea of her test, the idea behind them is that you could use a really small amount of like DNA or protein and run these experiments. But it was a beautiful idea that they weren't able to execute. But they were able to convince a lot of people they could. That is a very, if you guys haven't seen that Elizabeth Holmes, the inventor, that documentary, it's very interesting. Yes?
To add on, it's actually, they were diluting the blood too much, but when you dilute blood too much, it loses its, I don't remember the ways they say it, but it no longer is reliable, because you could dilute it with like a bigger sample, but if you're diluting it so much and running so much tests on like something that's unreliable, it loses the, really good.
0.01% of your blood cells have a certain mutation in them, but it's enough to give a disease. So you could collect those cells and collect out the DNA, and there might be only 1 or 2 copies of that DNA in what's collected, because you might collect a lot of healthy cells too. There's actually a way they look for HPV as well. So if anyone's ever had a pap smear and you get an HPV test, they collect a lot of cells, and maybe one of those cells has one piece of the HPV DNA. You would never be able to detect it if you didn't amplify it. So they actually do a PCR reaction on those samples, and they amplify. So you're building up the HPV part. They put primers around where there would be HPV gene, and then after many cycles, everything else that's healthy, you'd have just 1 or 2 copies. And the gene that's the sign of disease, like HPV, you'd have 1 million copies, so you could see it, so you could detect it. Does that make sense? So in diagnostics too, amplification is really important. Great, all really good answers. Excellent. So if there's any last questions, we can take those. But otherwise, you guys are set to go enjoy the rest of that. Thank you. Really excellent.